Now we will discuss in detail the functional aspect of class 1 molecules and after that class 2 molecules. So let's suppose this is one cell in your body and we see how class 1 molecule work there. What is their role in physiological role in immune system. Let's suppose this is a cell and here is its nucleus and here is your beloved chromosome number 6, one from maternal side, another from paternal side, and let's suppose this is class 1 gene box, right? A, B, C, and one of the gene expresses here. When one gene expresses here, naturally initially it is going to make messenger RNA, right? One of the class 1 gene has expressed itself and messenger RNA is formed. After processing in the nucleus, messenger RNA will come into cytoplasm. Now in the cytoplasm, you know, there must be endoplasmic reticulum. This is endoplasmic reticulum. Now if it is rough endoplasmic reticulum, you must be knowing that these are ribosomes and messenger RNA will come over here and come on the ribosomes and it will be translated here into proteins, peptide chain. So a peptide chain will be made here and this peptide chain will come into endoplasmic reticulum for further processing. This peptide chain is actually alpha chain of the class 1 molecule. Maybe it is A or B or C, but it is alpha chain of the class 1 molecule. When this chain will come in, it will undergo appropriate folding, and once it is folded, it will become a molecule with appropriate folding. It will fold it into 3 domains: alpha 1 domain, alpha 2 domain, and alpha 3 domain. Okay, let me make it here. Once it is there, then it will convert into alpha 1 domain, alpha 2, alpha 3 domain initially. Is that right? And this is its hypervariable region which is fit to bind a antigen. Meanwhile, you must be remembering that chromosome number, which one? 15 is also expressing, and this particular gene which expresses, it make another messenger RNA on the ribosome which will make another protein which will go in, and this protein is called beta 2 microglobulin. This beta 2 microglobulin will fold appropriately and then it will bind with the alpha chain. Now both of them are making together complete class 1 molecule. Now what is the function of class 1 molecule? One function we have seen it, that it is for identification of self and non-self tissue. But does it have any other function? As I told you that every cell, every nucleated cell is constantly making the stream of class 1 molecule. Every minute new class 1 molecules are being made. The only differences in those class 1 molecule, very important differences in every class 1 molecule is that these pockets may be varying from molecule to molecule. First molecule will have a different pocket, another molecule will be made after some time will have a different pocket, still another molecule will be made with this different pocket. So why this is happening so? We'll understand it more clearly when we talk about the intracellular protein synthesis. You know, every cell synthesizes a lot of enzymes and a lot of structural protein for its structure and function, right? Now there is a very interesting thing. Even though every cell synthesizes proteins, every nucleated cell synthesizes proteins, right? Let's suppose there is another protein which is being synthesized here. This is ribosomes and this is messenger RNA, and here is, suppose, a peptide chain which is being made here. You will be surprised to know that there are special type of cutters here. The special type of enzymes which act as biological cutters. Now, what really happens? Whatever proteins are synthesized within the cell, or whatever proteins are present within the cell, some of these proteins samples are cut into pieces by this enzyme. Again listen. For example, this cell synthesizes 1000 type of different proteins. Out of every group of protein, few peptide chain will be cut into pieces by this enzyme. Let's suppose that this chain passes through this and then it is converted into some peptide fragments. These peptide fragments, right, which are generated by this cutter, they are transported into endoplasmic reticulum by a special transporter protein. It means that every protein which is present or synthesized within the cell, its sample will be available in the endoplasmic, its cut pieces will be available in the endoplasmic reticulum. Is that right? Now you may be thinking, what is the purpose of that? Let me explain. First, what is the name of this cutter? This cutter is called proteasome. Proteasome. So we can say in every cell there are special enzymes called proteasomes which are cutting the samples of every protein into small cut pieces, and then there is other proteins which transport these cut pieces into endoplasmic reticulum, and these transporter proteins are called TAPs. TAPs, right? Or we simply call them transporter proteins for the peptide fragment. Now these peptide fragments will, let's suppose these are the peptide fragments of that thing. These peptide fragments will go here. Now while they are going into endoplasmic reticulum, somewhere they will have a chance to meet newly formed class 1 molecule. Now let's suppose this is one of the test fragments. So this fragment come here. Sooner or later, this peptide fragment of this protein, this peptide fragment will find sooner or later such class 1 molecule where this can fit into this position. Now listen, every minute, new and new pieces of, new and new samples of peptide fragments are coming here, and every minute new and new class 1 molecules are coming with slightly different pockets. Sooner or later, this peptide fragment will eventually find such class 1 molecule which has a such type of this pocket into which this peptide can fit. Once this peptide fit with in the pocket of alpha chain, then beta 2 also bind with this. This become trimeric unit. It has 3 peptide. Number 1 peptide fragment as a sample protein, alpha chain, alpha peptide of class 1 molecule, and beta 2 microglobin. Then when these 3 come together, they become stabilized. Then they pass through endoplasmic reticulum, through the Golgi apparatus, they are going to the cell membrane, and eventually they get planted into cell membrane. So what you really find there, that every class 1 molecule which is being expressed on the surface of the cell has a special type of, you can say, peptide in its pocket. Let's suppose this was the peptide it captured, and this peptide could fit into this pocket. Now listen, if 10,000 class 1 molecules are coming and expressing here, it is quite possible every class 1 molecule has slightly different peptide held into its pocket. So what is the real purpose of class 1 molecule? One of the very important and real function of the class 1 molecule is to present the samples of or fragments of peptides which are endogenously synthesized and endogenously present in the cell on the surface of the cell. Because our immune system players are outside the cells, not inside the cell. You know, different type of immune system components, they are present outside the cells. So every cell report on the surface the samples of its internal protein with the help of class 1, so that immune system should know what are the proteins which are being synthesized within every cell. Do you understand it? What is the function of the class 1 molecule? Class 1 molecules are the very secret reporters. What they are reporting? They are reporting on the surface of the cell about every peptide which is present or being synthesized within the cell, so that immune system is always updated that what are the different proteins present within the cell. Am I clear? Now, if there are many peptides, class 1 molecules, let's suppose I make 3 class 1 molecules here. Of course, all of them will have beta 2, yes please, microglobin. But these things may be different. It has a different type of peptide fragment. It has different type of peptide fragment, and this may have a different type of peptide fragment. Do you understand it? But all of them are class 1 molecules, so all of them will have this area same, which is for CD8 interaction. Is that right? Now, you can understand that in the beginning of the lecture, I said that one part of the gene of class 1 molecule is constantly shuffling. What that was doing? That was ensuring that different class 1 molecules have different antigen binding pockets, so that by chance, every peptide fragment should find at least one class 1 molecule with which it can bind and eventually get expressed in the cell. So fusion of peptide fragment with the class 1 molecule occur within the endoplasmic reticulum. And this class 1 system is a special reporting system, right, which report the fragments of endogenous proteins on the surface of the cell. Now what really happens? Every cell is visited by different immune cells. For example, I tell you natural killer cell. Have you heard of natural killer cell? Natural killer cell can recognize only your own class 1. My natural killer cell can only recognize my own class 1 molecules. And my natural killer cells are only happy with my own class 1. Is that right? If you bring, what happens? Natural killer cells come to this cell, interact with these molecules. If natural killer cell finds that this cell has self class 1 molecule, natural killer cell will be happy that, okay, this cell has my own self class 1, it means it's my own cell. So natural killer cell will not damage and move on forward. So every day, every moment, natural killer cells are going to different cells and ensuring that class 1 molecules present on those cells belong to the self category. And until any cell expresses self class 1 molecule, natural killer cell never disturb that cell. You are understanding? Now what happened? In grafted tissue, for example, you bring Lewis kidney and put into my body. What will happen? Lewis kidney cells, grafted cells, will have class 1 different than my own class 1. My natural killer cell love to be happy with only those cells which are decorated with my own class 1. So when my natural killer cell through circulation will go to the grafted tissue and they will study the grafted tissue class 1 molecule, they will find that they are sensitive to the presence of self class 1. They will find oh my God, what natural killer cell will find? They will find, oh my God, there are some cells and they don't have my own class 1 molecule. They will immediately destroy those cells. They will naturally destroy those cells. You are understanding? This is one problem, that grafted tissue, because it does not have the self class 1 molecules, natural killer cells simply destroy. As you think of a mother who loves, it's a mother who is weird, she loves only her own children, and if in her home she finds some other children, she will kill. She is naturally killing mother, right? But still, she will love to have her own babies. So natural killer cells love to be with their own cells. What are the clinical and immunological implications of this concept? The implications of this concept are, number one, grafted tissue does not, mismatching grafting tissue does not have recipient class 1 molecules. So recipient natural killer cell gets frustrated with the grafted tissue because it does not express appropriate class 1 and it attacks the grafted tissue. This is one implication. Another implication, sometimes some virus try to be clever. Some virus come into our body and they are very afraid that viral proteins will be reported on the outside the cell in association with class 1 molecule. Let's suppose there's a virus which enter into this cell. Normally what happens, when virus will synthesize the proteins within our cells, fragment of viral protein will get associated with class 1 molecules, and viral proteins fragment in association with class 1 molecules will be expressed on the surface of the cell. That is how our immune system will come to know, oh my God, this cell has some viral proteins in it, let's destroy the base of the virus. But some viruses are super clever. You know what they do? That's really genius virus, I believe. You know what they do? They produce some protein which go and switch off the class 1 genes. What a wonderful thing. Some virus produce some protein. HIV is one of those viruses. In some people, some HIV virus produce some special proteins which go and switch off which genes? Class 1 gene. And class 1 molecules are not synthesized. And if class 1 molecules are not synthesized, is that right? So of course class 1 molecule cannot report the presence of the virus on the surface. Is that right? So of course virus specific T cytotoxic will not attack that cell because the immune system will become blind to the presence of virus. Virus is very happy inside that I'm not being reported. But our immune system is a human being's immune system. Still it will manage the attack. You know how? Some natural killer will keep on dancing around, find the cell, say why class 1 are not there? Oh my God. He doesn't know that there is a virus in and playing their trick. Natural killer cell will think it is not my cell. Kill this cell. So still viral base will be destroyed. You are understanding? In the same way, sometimes happen in tumors, some cancer. If there is a cancer in the cell, then fragments of, listen, when a cell becomes cancerous, a cell becomes malignant, it may produce mutant proteins, altered protein. Chances are that in a cancer cell, when mutant and altered proteins are produced, those protein fragments may bind with class 1 and get reported. And then T cytotoxic may attack such cell which is having mutant proteins. But some cancer also sometimes try to be clever. In some malignant cells, they switch off their class 1 genes. Some cancer cells, a malignant cell switch off their class 1 gene. The benefit is that if class 1 genes are switched off, then cancer, the proteins associated with cancer cell will not be expressed on the surface. And immune system may not know that this cell is altered, having dangerous protein in, or it is transformed cell. Fine. Immune system will be blind to the presence of mutant protein as they are not being expressed on the surface with class 1. Right? But again, who will come into play? Natural killer cell. It will come to this cancer cell. It will try to find where are my class 1. It will not find there are class 1. bomb, kill that cell. Is that right? You will study in some other videos how natural killer cell kill, but they do kill. They do contact killing. They just do contact and kill. It's a very non-romantic thing, but they do it. So there's a lot of implication of clinical and immunological implications of this concept that all of our cells must express our own class 1 molecule so that our natural killer cell should remain happy with us and don't attack our normal cells. Is that right? Am I clear to you? Yeah. Opposite to that, whenever a cell does not express our own class 1 molecule, right? Whenever a cell does not express our own class molecules, cell will be attacked by natural killer cell. What are the circumstances in which our own class 1 molecules will not express class 1 molecule? What are the circumstances in which our own cells will not express class 1 molecule? I mentioned 3 conditions. Number 1, when special virus switch off the class 1 genes. Number 2, when certain malignant cells switch off their class 1 genes. Number 3, when transplanted mismatching cells are put into our body and our natural killer cell don't find self class 1. This is one whole application of the concept of class 1 molecule. Another application. As I told you that class 1 molecules are constantly taking the fragments of our proteins and expressing on the surface. Let's suppose this is our self protein. Let us suppose that this is our own protein. Now, we talk about T cytotoxic cell. T cytotoxic cells. You know, T cells have special receptor to recognize the antigens, and those receptors are called TCRs. T cells have TCRs. Let's suppose this is a TCR of this T cell. Now here you have to remember one thing. In the early videos of the immune system, I explained that all those T cells which make such TCR which could react with our own proteins, those T cells are destroyed during the early development of the immune system. Again, listen carefully. When our immune system is building, our own proteins are offered to the T cells. And if by chance any T cell has made such TCRs which could engage with our own proteins, that T cell is destroyed within the thymus. But those T cells which are making such TCRs which do not bind with our own proteins, those T cells are allowed to go into circulation. So it means right now my body has a pool of T cells which have such TCRs which can react with any antigen in the peptide antigen in the world, but they cannot react with my own proteins. Are you understanding? In every person's pool of T cell, in every person's collection of T cells, such T cells are missing which could have TCRs which could recognize that person's antigen. That is why our own T cells tolerate our own proteins and our own tissues. That is related, this concept is related with the concept of self-tolerance. One of the reasons why our immune system tolerate our self-proteins and self-tissues, simply because such T cells which had such TCR which could interact or engage with our own proteins have been destroyed in the early development of immune system. Now imagine, this is a normal self-protein, and this normal self-protein is expressed on the surface of this cell with the self class 1. This is T cytotoxic cell. When this T cytotoxic cell will come here, its TCR can engage with our own protein? Do you think our T cells have such TCRs which engage with our own protein? No. So when TCRs will come to study the surface of the cell, this T cell will not have such TCR which could engage with our own peptide fragment. So this T cell will not engage with this cell here. But let's suppose this is another class 1 molecule which is having a different peptide fragment, and let's suppose this peptide fragment is from a viral protein. Let's suppose there was a virus in, and that virus was making its own proteins, and let's suppose that this viral protein fragment has gone in and with class 1 has come here. Now this viral protein is a foreign protein. Sooner or later, one T cell will come over here which has such TCR, which can, this is a TCR of a which cell? T cell. Sooner or later, one T cell will come with such TCR which can engage with this viral antigen. As soon as it will engage with this, right, so this cell will get report that one of the protein which is expressed on the surface of this cell is foreign to us. So this cell will get a report that within this cell there is something fishy. There is something which is not alright. Probably there are some proteins which are altered or foreign proteins. So then immediately this will engage its CD8 molecule with constant domain of class 1. What it is doing? It is confirming, is it our own cell or not? If TCR is engaged with the protein peptide fragment and CD8 is engaged with the class 1, if both of them are appropriately engaged, they give intracellular signals. They give signals through the CD3 molecule. TCR and CD8 molecule activate CD3 which gives signal to the cell. What is the signal? The signal to this cell is, signal to the cytotoxic cell is, hey, I have found a cell, my own cell, having a foreign peptide or altered peptide in association with class 1 molecule. Then this T cell will be activated. So when these T cytotoxic cells are activated, T cytotoxic cells are only activated when peptide fragments are expressed on the surface of the membrane in association with class 1 molecule. So through CD8 component, they confirm the presence of class 1 molecule, and with the help of TCR, they recognize the peptide fragment, and after that this T cell will become activated. Right? In early videos, you must have studied once T cytotoxic is activated, what it is doing. Number one, it will release a product called perforin. Perforin. These are special proteins which are present within the T cytotoxic, and once T cytotoxic cells are activated, they release perforin proteins and they will come on the surface of this target cell, and perforin proteins will assemble, these proteins will assemble into special type of pores. These are perforin protein. Let's suppose this is one perforin protein, this is another perforin protein, this is another perforin protein, here is another perforin proteins. These proteins are released by T cytotoxic, they assemble on the membrane of the target cell and make a hole. In a way, it has perforated the target cell, which was having some suspicious antigen, and which, right? And of course, what will happen? Internal substances will go out and external substances will go in. Membrane integrity as permeable, semi-permeable membrane is lost. Structural integrity of the membrane is lost. So it is just like the perforins come like a drill and they drill the holes into target membrane. When multiple perforations occur, do you think this cell can survive? So this is one ruthless way how T cytotoxic, why we call it cytotoxic, because it produces toxicity in the cells, target cell, when it is activated. They release the perforins and they produce perforations and hole in the target cell. But after that, this is cruel to produce perforations. After that, they do something very mean. You know what they do? A very mean activity. If you want to kill your enemy, number one, you produce a drill holes. It is bad. But if you're really mean man, what you will do? In his wound, you will put some chili and salt. It's a very cheap, mean activity, isn't it? This is what T cytotoxic does. You know what it does? It has another special type of proteins, and it will release those proteins, and through these perforations, those proteins will go in. And these proteins are very dangerous enzymes, and they'll start digesting many internal proteins, and even these proteins like chili and salt will do further damage, they may activate the pathway of apoptosis. Is that right? These proteins are called granzymes. Granzymes. Right? Still it is not satisfied. Most of the cell express Fas receptor. Fas receptor. And this may engage its Fas ligand on the Fas receptor, and this may also give signal for apoptosis. And eventually this cell in which virus was there, it will be destroyed. But all this game depends on the presence of class 1 molecule appropriately expressing on the surface of the cell with suspicious fragment of a peptide, right, in its pocket. Is that clear? So viruses are really in trouble. When virus come here, if they let the class 1 molecules work, class 1 molecules are very mean. They take the fragment of the virus and express on the surface so that T cytotoxic can attack. And if virus try to be shrewd and switch off the class 1 molecule, then the natural killer cell come and destroy the cell because they find there are no class 1 molecules. Is that right? Am I clear? Now this concept which we have discussed in relation to class 1 molecule, it is very important in transplant, it is very important in tumor immunology, and it is very important in, yes, malignancy and immune system. Let me relate again. Number 1, if you transplant for example Lewis kidney in my body, now kidney may get two type of attack. Number 1, the grafted tissue does not have my self class 1 molecule, so natural killer will attack. Plus another trouble, grafted tissue will express class 1 molecule on the grafted cell with the peptides which are foreign for me, because if you take Lewis kidney and put in my body, is that right, then transplanted cells will express the Lewis class 1 molecule with those proteins which are synthesized in Lewis body, not Najib's protein, not my protein. So another way why graft will be attacked, because graft is expressing class 1 molecule, right, with Lewis protein, not with my protein, and my body has T cytotoxic which will recognize Lewis protein as foreign protein and attack that. So it means this class 1 concept is related with transplantation, right? Ideally the transplanted or grafted tissue should have the same class 1 molecules as is the recipient class 1 molecule. Is that right? Clear? Yes.
So only T cells and natural killer cells are involved in
transplant rejection. There are other mechanisms also, we'll talk later, right? This is one of the mechanisms. Second concept is related with tumor immunology. When there is a malignant cell, it develops many mutations and every cell makes a lot of protein. So malignant cell also makes some proteins, but those are mutant proteins. Those mutant and altered proteins fragment may be expressed with the class 1 on the surface of cancer cell. So these mutant proteins fragment with the class 1, when they express on the cancer cell, our immune system take those mutant proteins as foreign proteins present in the cell. So our T cytotoxic cells will attack the cancer cell which are expressing the altered mutant protein. Could you understand it or not? If you have not understood, I can make it more clear. You are not sure. I should make it more clear. Okay, listen. When a cell becomes malignant, then its genome becomes unstable. In its multiple genes, mutations develop. Now listen, when cell was healthy, it was producing normal self class 1, and in the mouth of self class 1, when cell was healthy, it was having the fragments of endogenous normal proteins. And when cell is expressing normal proteins, T cytotoxic don't interact. Is that right? Because T cytotoxic cells do not have TCRs for normal proteins. Is that clear? No problem into this? Fine. Now you imagine this cell become malignant. This cell has to make many of its normal enzymes and structural proteins. Some of those normal enzymes and structural proteins are now mutant. So those fragments of those mutant proteins will be expressed on the surface of the cell with in association with class 1 molecule. Is that right? Now cancer cell may be expressing the self class 1, but in their pocket they will have mutant proteins. Is that right? Whenever T cytotoxic will come, some of the T cytotoxic may have TCR against the mutant protein, and those T cytotoxic will attack the cancer cell. Is that right? So it means that class 1 molecules, by expressing the cancer peptides on the surface, help us to help our immune system to identify the malignant cells and help the immune system to kill the cancer cells. Is that right? In the same way, class 1 molecule also help our immune system to recognize the viral proteins or other microbiological proteins which are present within a cell. Is that right? No problem. So I was saying that this class 1 molecules concept help you to understand what is the role of these molecules in the grafted tissue and matching and mismatching. It also explains what is the role of these molecules in malignant cells, that they can express the altered protein. But normally these molecules are expressing fragments of normal protein against which our immune system normally does not react. Am I clear? Anyone has any question here? Right. So now we come to this thing that class 1 molecules have how many functions? Can you tell me how many functions they have? Number 1, they are for self-recognition. Self-recognition. Right? And number 2, they are playing a role in reporting of endogenously synthesized proteins to extracellular environment where T cytotoxic are there. So that if within the cell there is viral protein or abnormal bacterial protein or abnormal malignant mutant protein, class 1 molecules help all those things to be reported on the surface of the cell so that T cytotoxic can take notice of that. Am I clear? So this was something about class 1 molecules. Let's have a break.